Hello everyone, I am Alexis Halvin, and today I'm going to be talking to you guys about real-time polymerase chain reaction, also known as PCR. What is PCR? PCR is a method used to analyze a short sequence of DNA or RNA. So what you're going to do is you're going to take a small section of DNA or RNA, and then you're going to amplify it. So it's making a small section of DNA and amplifying it and making millions of copies. That's the quick overview of what it is. In comparison to DNA sequencing, that can take a few days, it's a lot less time consuming and it's pretty inexpensive. So in 1983, Kary Mullis was an American biochemist who later won a Nobel Prize in 1993 for this development of PCR. This is just a quick overview. We're going to get more into it here in a little bit. I'm not going to go too in depth about him, what he did, and all that stuff, but it is really interesting how he did it and why he did it. So what is PCR used for? There are three large categories in which PCR is used. It's used to understand the function of genes to match crime scene DNA, and then it's also used in biology and medicine. We're going to talk a little bit more in depth about these here in a little bit, but I just wanted you guys to see the importance of PCR and what it might be used for. Before we get into PCR, I just want to really emphasize the fact that DNA replication is key to understanding PCR. So before we go into understanding PCR and how it works and all that stuff, we're going to do a quick overview of DNA replication. So first we're going to talk about what makes up DNA and how that would be important in understanding the DNA replication. So here we're going to go 1, 2, 3, 4, and then 5. The reason why I numbered this sugar here will be important in 1 second. So you have a sugar, a nitrogen base, and a phosphate group. You have two different types of bases. You have purines and pyrimidines. And then over here I just showed you what bonds with what. Two purines cannot bind together because it's going to be too bulky because they're too ringed. And then the pyrimidines cannot bind together because it'll be too thin. So you're going to need a purine with a pyrimidine. And then in RNA, there is no thymine, there's only uracil. So I just thought I would put that in there so you guys knew. How I number these carbons over here, this is important in knowing the direction in which DNA is replicating. So the original strand is always replicated, it's always read in the 3 to 5 prime direction, and then it's added in the 5 to 3. So you can see that at the third carbon, it's added to the fifth. The third carbon is added to the fifth. Third carbon is added to the fifth. That is why it's the 3 to 5 prime direction, and then its complementary strand is 5 prime to 3, 5 to 3. So then you would say that this strand is being added in the 5 to 3 prime direction. Over here is just a linear diagram going 5 prime to 3 prime as the new strand and the original strand as 3 to 5. Looking at this once again in another diagram, we see that the old strand is a 3 prime to 5 prime and that the new strand is being added 5 prime to 3 prime. Because of that, when you look down at the second diagram, due to DNA replicating from the 5 or the 3 prime end to the 5 prime end, the second strand of DNA, that is 5 prime to 3 prime, must be replicated in segments referred to the Okazaki fragments. And that is just because it is going in the opposite direction that DNA naturally replicates in. Again, here's just a little overview. We know that DNA is a double helix, so the helicase comes in and unwinds that double helix. This enzyme over here is going to relieve the torsional strain from the helicase. You have a primase, which initiates the synthesis of an RNA primer. I know it kind of sounds weird that an RNA primer is used in DNA, but they're extremely similar, and then a DNA polymerase just comes in, takes out the primer, and sticks in DNA, and then continues the synthesis. Now, the DNA polymerase 3 is more predominantly found on the leading strand, whereas DNA polymerase 1 is more predominantly found on the lagging strand. And with this lagging strand, like we talked about a second ago with the Okazaki fragments, these fragments need to be binded together, and that is what DNA ligase does. So one last little overview of this. These right here are hydrogen bonds in between the C's and G's and A's and T's. So those are going to get broken, it's going to get opened up, and it's going to get replicated. From there, we can see over here that it is considered, that DNA replication is considered to be semi-conservative, and that is because you have two parent strands, and then it gets opened up and replicated, so now you have one parent strand and then one new hybrid. And over here you have one parent strand and one new hybrid. So that is just something to consider. So now that we know how DNA replicates, we're going to discuss how does PCR use DNA replication. But before we get a little bit more into that, we're going to first talk about the ingredients that are needed for PCR. So you're going to need a DNA template, primers, DNA polymerase, dNTPs, which are your free nucleotides, and your buffers and cofactors. Your buffers and cofactors change every time that you do PCR. It's very specific to what you're trying to find within the DNA sequence. So all of this gets put into a test tube, and then right here, this Taq DNA polymerase, that is actually pulled out of a thermal bacteria, and the reason why is because here in a second you're going to see the different stages that this needs to go through, and one of them is a very hot temperature, so you need a DNA polymerase that can withstand all of that heat. These are the three main stages. The first stage is denaturing. So what's going to happen, instead of the helicase coming in, because that's naturally occurring, we have to physically open it up. So we're going to heat it up to about 94 to 96 degrees Celsius. We're going to break those hydrogen bonds and have two single-stranded templates now, remembering that DNA is naturally a double helix. So then we're going to cool back down a little bit in the annealing phase, and then those are going to allow the primers to bind to its complementary strands. And then extension, we're going to heat it up just a little bit, and that is so that the Taq polymerase extends so that primers can now synthesize the new DNA strand. And then the cycle just continues. So I'm going to show you what we actually did in our lab. So this is a little overview clips from the lab procedure that we did. You can see the ingredients. We had water, a buffer, magnesium chloride. The magnesium chloride is used so that the magnesium ions allow for the reaction between the hydroxyl group on the new strand and the alpha phosphate on incoming dNTPs. For some people, that might be over your head, but for some people that were curious, that is the explanation for that. You have the free nucleotides, the primers, the Taq polymerase, and then the sample of DNA. These were the quantities that we used, and now over here is how long and for what temperature that we did each cycle. This is what the PCR does. So the PCR is going to go up to 95 degrees for one cycle, and then it's going to go 95, 63, 72 each for 30 seconds, and that cycle happens 35 times. That is so that each section can get amplified and amplified and amplified, so you have thousands upon thousands of copies of that small section. And then the final extension, you have 72 degrees Celsius for 10 minutes and then 4 degrees Celsius until you pick it up. So these are kind of the quick overview of the cycle, what temperature you're talking, and remember, these are degrees Celsius, so it's not degrees Fahrenheit, it's a lot hotter than it looks. So it's just something that was really interesting to see that this happens 35 times and why it happens like that, and that each one of these stages requires such a different temperature. So why is this important? So for DNA mapping, one for the Human Genome Project, that was when it was an international effort to determine the base pairs that make up human DNA. The PCR played a really important factor in that. You have genetic testing for potential disorders, whether it be chromosomal or just other type of disorders that somebody might be testing for. They can go in, pick a certain section that they know this disorder is predominantly found, and will test it. Personal DNA testing. I know a lot of people have heard about Ancestry and 23andMe. This is my actual DNA. I got it tested in like 8th grade. So that is what I made up of. And then for forensic, a small sample from a crime scene can be amplified and compared to suspect's DNA. So it has been able to solve cold cases and clear wrongfully convicted people. I found that really interesting, and if anybody watches like criminal shows, you might have heard PCR, like that phrase and that word been thrown around. that was something that I found really, really interesting too. So PCR and COVID-19. So before we get into COVID in particularly, anything that has a viral genome is actually an RNA. So like I said earlier, PCR works for RNA or DNA, but it needs to get from RNA to DNA. And how that works is it goes through the process of adding an enzyme called reverse transcriptase, and that then allows it to be read as DNA, and then it can go through that process. So that's what happens with anything that has a viral genome, such as COVID-19. So it works by detecting specific genetic materials within the virus. There are different samples that can be taken, such as a swab of the throat, saliva, liquid from the lower respiratory tract, or a stool sample. Then the nucleic acid gets pulled out of that, and then you amplify specific regions. Now, the University of Washington School of Medicine chose 100 nucleotides that were very specific to the virus, and within that 100 nucleotides were codes for 2 very specific genes for the virus. So a lot of other tests have found very similar things, and that is how they're using PCR to determine whether somebody has the virus or not. So with the results, it's positive if the results show that both genes are present. It's inconclusive if only one of the genes are present, and then it's negative if neither one of them are present. With pros, there are cons. Like we talked about earlier, it's quick, it's inexpensive, and it detects the actual virus, but there are cons because it is extremely sensitive. You're adding so many different ingredients within that test tube. If you add too much or too little of something, there could be an issue. So it's very sensitive and false results can happen. And you're only able to use it if the patient is currently sick with the virus. So if the patient is recovering from the virus and no longer physically has it within the body, you're not going to be able to see it because it's detecting the genes. So if it's detecting the genes of the virus, you need to physically have the virus. So blood samples, when you're using an antibody test, it will tell you if the person has ever been exposed, and that's because those antibodies will continue to be in your body even when the virus has been killed off. So with the antibody test, you don't have the liability that PCR has when it comes to the results. Alright, that brings us to the end. I hope you guys enjoyed this presentation and took something away from it. And for some people, maybe it sparked an interest to learn a little bit more about PCR and COVID-19. Thank you all for staying till the end. I truly appreciate it.